Hello, everyone. I want to first start by thanking the Bionano team for this invitation to speak in this symposium. My name is Madhuri Hegde. I am the Senior Vice President and Chief Scientific Officer at Revvity. And today I'm going to talk about molecular analysis of FSHD using optical genome mapping. So, as we all are very aware that whole genome sequencing, even though with the tremendous advances that have happened in the past decade or so, there are still many disorders that cannot be detected using whole genome sequencing. And I have just laid out some challenges here in the box in green, which talks about the repeat disorders. Some of the larger repeat disorders, for example, Fragile X, can still remain challenging. Larger and complex repeat segments, which we are going to talk about today, FSHD1, balanced translocations, chromosomal rearrangements, and methylation disorders. It's given that the technology is advancing very quickly, and it's really interesting that today there are some technologies that which we can bring into our laboratories to detect some of these complex disorders, such as FSHD. There are three technologies I've highlighted here: digital genome mapping, long read, and sequencing by expansion. And today I'm going to focus this presentation on digital genome mapping. So just as a background to Revvity Omics, we are a global laboratory, and by that I mean we have a global footprint. What you can see on this slide is our main laboratory is in Pittsburgh in the US, followed by Interpretation Center in Atlanta, Viacom cord blood and banking service in Hebron, and then our global laboratories in UK, in Manchester, Cambridge, Oxford, Sweden, India, and China. The reason I show this slide is really this global footprint really allows us to test cases from all over the world and thereby really develop a very deep database of the different variants and improve variant interpretation. And we really leverage our global footprint in when we started validating the assay for FSHD1. We do a broad range of testing. Revvity, formerly PerkinElmer, is really known for newborn screening, and then it expands from newborn screening into whole genome sequencing, exome sequencing. Copy number genome is a low pass genome. Neuromuscular disorders panels is highlighted here because it is relevant for the FSHD testing. And then we also complement our neuromuscular panel with FSHD testing using the Bionano genomic Sapphire as a tool of choice to validate our LDT. So let's jump right into it for first talk about what is FSHD. Some facts about FSHD: it is the third most common muscular dystrophy in the general population. And the incidence is really not that rare, about 1 in 8,500 worldwide. There are two types of FSHD, FSHD1 and FSHD2. The overall symptoms of FSHD are quite broad, starting with scapular winging, facial weakness, atrophy of the upper arms, abdominal weakness, lower leg weakness, chronic pain, retinopathy, and high tone sensory neural hearing loss. The reason I read out this clinical presentation to you all is that FSHD1 and FSHD2, though clinically similar, their molecular events are different and very interesting, and we'll talk about that in a little bit. It's not easy to molecularly separate FSHD1 and 2, and this is where the power of the optical genome mapping comes into play. So let's look at FSHD1 and 2. FSHD1 is the most common autosomal dominant, about 95% of the cases. And a very small number of cases, about 5% of the cases, are FSHD2. And the inheritance is digenic dominant inheritance. We are going to talk about that in a little bit. Cases with pathogenic variant in chromatin modifier genes such as SMCHD1, DNMT3B, and LRIF1 have profound hypomethylation of chromosomes 4 and 10, which really lead to the toxic expression of the DUX4 transcript on chromosome 4. So let's look at the mechanism a little bit. On this right-hand slide, you see a figure. The blue triangles are the closed chromatin, orange are open chromatin. The DUX4 gene is embedded in the D4Z4 repeats on chromosome 4. Essentially, what happens is the contraction of the D4Z4 repeats leads to the toxic expression of the DUX4 gene and thereby the clinical presentation. Now, this is on the chromosome 4, and there are two alleles, 4QA and 4QB. The D4Z4 repeats are also present or have a homologous sequence on chromosome 10. So now you're starting to see the complex picture here. The 4QA allele is a permissive allele. 4QB and 10q25 are non-permissive. They do not have a polyadenylation signal. So the permissive allele is 4QA, and the contraction on the 4QA allele is what is leading to the expression of a stable RNA of DUX4 and thereby the clinical presentation. So any assay which we develop has to be able to distinguish between the two alleles, 4QA and 4QB, and also 10q25. Now, the three diagrams below the normal repeat size, which is shown above, which is 11 to 100 of the D4Z4 repeats. The first one is for the contraction of the 4QA allele, which leads to expression of the DUX4. And then the FSHD2, which is the transmutations which act as epigenetic modifiers and thereby reduce the methylation or cause hypomethylation and thereby expression of the DUX4 gene. So this is a very complex mechanism that leads to the expression of the DUX4 gene. Now, in the last several years, there have been many publications of different ways to look at this D4Z4 repeats and how to actually measure them. There are complex methods that have been developed in the literature as well. What we really focused was on using optical genome mapping in our laboratory to develop an assay to distinguish between FSHD1 and FSHD2 and thereby have a LDT in our laboratory. So you all are very familiar with this slide, and I'm not going to repeat it here, but essentially we are trying to discern these alleles using optical genome mapping. Also, what was really important to us is that we have a workflow which also gives us analysis tools in our laboratory so that everything can be compiled into one single assay. So the idea being that we start with isolation, followed by labeling and loading, and then analysis. And obviously, turnaround time is very important in any laboratory, and thereby we achieve the completion of the assay in a very desirable turnaround time. So what does the optical genome mapping allow us to do? The first thing, as I mentioned in the previous mechanistic slide, is it's really important to distinguish between the 4QA and the 4QB allele, because the 4QA allele is a permissive allele. So the optical genome mapping allows us to distinguish between these two haplotypes. Then confidently discern between the 4Q and the 10QD4Z4 arrays. Measure the D4Z4 repeat on chromosome 4, but then also some additional requirements such as reporting of the structural variants or the copy number variants proximal to D4Z4 is important. Detecting copy number variants in these other three genes which I mentioned, and the most important of them being the SMCHD1, and then produce a comprehensive result, which then including images which they can translate into the report which we are going to eventually send out. So we have done extensive amount of work in the past 3 years on the optical genome mapping, and we were able to publish this paper in Neurology Genetics. I want to highlight some of the authors here, which really reflect to our global footprint. So Richard Lammers and Van de Merel are the really represent the story of D4Z4 allele, where they actually discovered this mechanism behind DUX4 gene expression. And Dr. Satish Khadilkar in Mumbai, who was able to provide us samples for validation, and he continues to send us samples for FSHD clinical assays. So let's look at the validation data. And the first and the most important thing is to compare the optical genome mapping method with the traditionally used method, which is a Southern blot method. This was step number one before we actually even go further. And what you can see here is that we were able to obtain these cell lines and compare the Southern blot method to the Revvity's optical genome mapping method. But we also went a step further as we collaborated with the Bionano Genomics lab, and we sent them the samples for a side-by-side comparison. And you can see the results on this slide that we were able to observe exactly the same results across the board when we use this technology. You see the slight differences of one repeat higher or lower, and that is because of the method used to calculate the number of repeats. And this is very important to note here because that's the algorithm we have put in into our laboratory. Now, going further from that, as I mentioned, Dr. Satish Khadilkar, who's a neurologist in Mumbai, was able to send a sample to do the further validation on actual cases that he has diagnosed to have FSHD. So what you see here on this slide is a discerning of the allele 1 and 2. So you expect an allele which is a contracted allele on the 4qa haplotype, and the 4qb allele which is going to remain in the normal repeat range. Again, what you see here is on the side-by-side comparison of optical genome mapping to Southern blot analysis, at the repeat size on Southern blot is one repeat higher, and that is due to the calculations that go into place to calculate the number of repeats. But that is already sort of programmed into our system. So how does that look visually? That is also important because as the data comes out, it's really important to visualize the data. So what you see here on this slide is the comparing it to the reference data and then putting the cases with their repeats on the same image. What you can see here is on the 4qa haplotype, the reduction of the D4Z4 report units to under, as I mentioned, 11 to 100 is in the normal range. And then the D4Z4 on the 4qb haplotype. So with that explanation, let's look at some cases. And we have really detected some very interesting cases in our laboratory. This particular case demonstrates that the power of optical genome mapping to detect mosaicism. So again, what you see here is a detection of one repeat unit, 8 and 12 on the 4qa allele, which is a permissive allele. And then on the 10qa we were able to detect, we were able to separate that from the homologous sequence of the D4Z4 on the 10qa allele, 10qa background as well. Now, obviously, the repeat size of 1 and 8 points towards FSHD1, and we were able to detect mosaicism in this particular case. Going on, as I already mentioned, that we are talking of autosomal dominant condition here, but in this particular case, we were able to detect biallelic D4Z4 repeat contraction, and what you see here is 6 repeat units and 9 repeat units, again on the permissive 4qa allele, that is then pointing towards FSHD1. Now let's move on to FSHD2. As I mentioned previously, for FSHD2, there are these three genes which cause hypomethylation and thereby expression of the DUX4 gene against a 4qa background. In our cohort, as shown on this slide, the FSHD2 cohort has a repeat size of 8 to 18. And we have detected several individuals now with a pathogenic variant SMCHD1 gene, and then thereby then going on to looking at the 4q alleles using optical genome mapping. Now, I want to show one interesting case here, and this is really important because why segregation matters, why discerning of these alleles is very important, is really demonstrated in this particular family here. So what you can see here up at the top is that the father is affected here. He has a nonsense pathogenic variant in the SMCHD1 gene, and two of the children are also affected because the SMCHD1 nonsense variant has segregated with the 4QA allele. In contrast, in this third child here, even even though the nonsense variant has segregated, it is not against the right background, and thereby this child is not affected. Now, this level of analysis is absolutely critical when you are doing familial analysis and actually trying to discern between the affected and the unaffected. Actually, at times we get pre-symptomatic testing in these families. So this kind of assay really helps you triage these cases appropriately. So in summary, we have had about 547 cases sent to our laboratory, and 308 were positive for the D4Z4 contraction on the 4QA allele, resulting in the molecular characterization of FSHD1. As I just showed, we have been able to detect mosaic alleles with at least one contracted 4QA allele, and that was observed in 9 cases. And 9 is quite a significant number here. A biallelic contraction of the 4QA was observed in 2 cases, and an apparently homozygous contraction was observed in 4 cases. We have also seen 9 cases of FSHD2. So what we have recommended in our manuscript, the published manuscript I showed before, is a combination of the NGS gene panel with OGM for testing for FSHD. Now, one thing I would highlight here is that we are not the first laboratory to demonstrate the use of optical genome mapping for FSHD. Now, there are at least 3 manuscripts which establish the efficacy and the sensitivity of OGM over other methods for FSHD. So we are essentially going through a combination methodology to look at these cases. So just finishing off, if you look at the very high level of what kind of algorithm can be put in place by a laboratory for molecular analysis of FSHD1 and 2. We're starting with optical genome mapping as a first step. If it is positive for contraction of 1 to 10 repeat units on the 4QA permissive allele, then there is nothing else that needs to be done further. If it's a carrier of a 4QA allele which has got more than 11 repeat sizes, we are saying, then sequence and del dupe the SMSCHD1 gene, and the other 2 genes can also be looked at. And then go on to issue a positive report for the PLP for the SMCHD1 gene. Then if you have no variant in that gene, then methylation testing becomes important. Now, if there are 2 4QB alleles, and it's not positive for contraction, then it is negative for FSHD, and a neuromuscular disease gene sequencing panel can be recommended after OGM has been finished. In cases of clear FSHD phenotype, and methylation testing can also, if you can see here that methylation testing can be recommended to rule out hypomethylation that may be caused due to complex D4Z4 alleles, such as rearrangements, unusual alleles. So we have this well established in our laboratory, and this algorithm has been published in our manuscript in neurology genetics. Obviously, any assay validation requires a large team, and I already mentioned our global footprint with clinicians, Dr. Satish Khadilkar, and the two individuals in that laboratory there, Rashna Dastur and Pradnya Gaitonde, who meticulously collected these samples with the clinical information and sent it to us. And the Revvity Omics team, led by Naga, who have really been very involved in not only validating this assay, putting this in clinical use, and really focusing on the details of how this data is getting represented and detecting this variety of different mechanisms from mosaicism to biallelic contraction. And we remain grateful to the Bionano Genomics team. They have always been available and open for discussions as we have continued improving and developing our assay. With that, I'm going to stop here, and thank you all for listening to this presentation, and I'm open to taking any questions. Thank you.